I think it's time to get started. So good morning. Thank you for attending today's workshop on the pathogenesis of CF airway infection. My name is Jamie Hook. I'm an assistant professor of medicine and microbiology at the Icahn School of Medicine at Mount Sinai in New York, and a chair for today's workshop. And to my left is Christine Rumpf, my co-chair for today's workshop. Christine is a PhD student at the Institute of Medical Microbiology at University Hospital Münster in Münster, Germany. Christine and I put together today's workshop with the goal of highlighting recent findings that advance our understanding of the host-pathogen and pathogen-pathogen interactions that promote airway infection in CF. We selected abstracts that bring out emerging themes in CF infection research, such as how antibiotic exposure and bidirectional interactions between host and pathogen promote lung infection, and how interactions between pathogens of different kingdoms shape lung infection development. We hope you find the presentations interesting, and we encourage you to ask questions after each presentation by speaking at the microphone or submitting questions in the meeting app. Each speaker has 15 minutes for their presentation and 5 minutes for questions. Christine and I will do our best to keep the workshop running on time. So with that, I'd like to introduce our first speaker, Juliana Donahoe. Juliana is a PhD candidate in the Baumberger Lab at the Giesel School of Medicine at Dartmouth. Her research focuses on how metabolic changes in the airway during viral infection influence secondary bacterial infections in people with CF. Please join me in welcoming Juliana.
Good morning, everyone. So thank you for that introduction. I'm happy to be here. So as they said, my name is Juliana Donahoe. I'm a third-year PhD candidate in Jen Baumberger's lab at Dartmouth, and I'm really excited to be here today. It's my first presentation at NACFC, and I'm excited to share with you my work on how oxidative stress and metabolic dysregulation can promote Pseudomonas aeruginosa biofilm growth during co-infections in CF. I have nothing to disclose. So over the course of their lifetime, people with CF will acquire numerous acute respiratory viral infections, similar to people without CF, but the consequences of these infections can be very severe. In fact, almost half of all pulmonary exacerbations in both children and adults with CF are associated with acute respiratory viral infections. And as you can imagine, this can have severe clinical outcomes for these patients, including increased hospitalization rates and prolonged hospital stays, as well as increase in antibiotic use, and then sort of concurrently an increase in respiratory symptoms paired with a decrease in lung function that often persists past the infection, and many patients never fully recover their lung function. And so viral infections on their own are a major consideration in the CF community, and this is compounded by the fact that viral infections are also associated with the acquisition of secondary bacterial infections. And so more specifically, RSV infections are associated with the acquisition of chronic Pseudomonas aeruginosa infections, and Pseudomonas is one of the most prevalent bacterial infections in CF and is a major contributor to the morbidity and mortality associated with CF. And so trying to better understand what interactions are happening between the viruses, the host, and Pseudomonas are a major consideration in the Baumberger lab. And so we can study this in vitro using this epithelial flow cell model shown here, where we can grow CF airway epithelial cells, called CFBEs, in a polarized manner and infect them with RSV and subsequently inoculate Pseudomonas and grow them under flow to assess biofilm growth. And as you can see in these images here, where the nuclei is stained in blue and the green is GFP-tagged Pseudomonas, when we have a preceding RSV infection shown in the right image, you can see that there is an increase in Pseudomonas biomass over 6 hours of growth, and that's quantified in the bottom right corner, indicating that we do see in vitro what happens in the clinic. And we know that this is not specific to just RSV, as if we do similar infections with human rhinovirus or adenovirus, we still see increases in Pseudomonas biofilm growth, as shown in the black bars. And we also see similar increases when we stimulate these cells with interferon, which stimulates the antiviral response that would be induced by viruses. And so all of this data together indicates that it is not necessarily a specific viral bacterial interaction, but that the viral infection is inducing changes in the host epithelium that is promoting these secondary bacterial infections. And so to look deeper at what's happening during these viral infections, we focused in on RSV infections and conducted proteomics. And so what I'm showing you here is pathway enrichment in CFBE cells during RSV infection. And unsurprisingly, there are a number of antiviral pathways that are being induced. And interestingly, we also see antioxidant pathways being enriched during RSV infection. And this turned our attention towards reactive oxygen species, or ROS. And so ROS is known to be generated during viral infections in both CF and non-CF airway epithelial cells. And they're also really key contributors to the induction of antioxidant pathways. And so going back to our proteomics, now looking on the protein level, I've highlighted here during RSV infection some of the most highly upregulated antiviral proteins in green, and then some of those antiviral or antioxidant proteins that are contributing to our antioxidant signaling pathways. And interestingly, when we treat these RSV-infected cells with a mitochondrial ROS scavenger called mitotempo, while we still see those antiviral proteins being significantly increased, we lose that antioxidant pathway signaling, indicating that the production of reactive oxygen species are responsible for and stimulating the antioxidant pathways that we see during viral infection. And this was interesting to us for a few reasons. Primarily that antioxidant production is typically related to metabolism, and we know that metabolic changes are being induced during viral infection. So in a normal uninfected cell, they're taking up glucose and sending it through glycolysis to generate pyruvate, which is then primarily being sent into the mitochondria and the TCA cycle to produce energy through oxidative phosphorylation. But during a viral infection, this metabolic state is shifted away from oxidative phosphorylation towards what's known as the Warburg effect. And in this environment, glucose consumption is increased in the cells, which are then still sent through glycolysis to generate pyruvate, but this pyruvate is primarily being converted into lactate instead of going into the TCA cycle, which is then being secreted out into the apical environment where Pseudomonas can interact with it. And in addition to this, there's also an increase in glucose 6-phosphate, one of the first components of glycolysis, being shunted away from glycolysis towards the pentose phosphate pathway, which is really important for the production of NADPH and antioxidants. And so we hypothesized that potentially these reactive oxygen species could be contributing to this metabolic shift during viral infection. And so to look at this, we looked at two of the main readouts of the Warburg effect, glucose consumption and lactate secretion. And so as you can see here, during RSV infection, we see increases in both. And when we treat these CFBE cells with the mitotempo ROS scavenger again, we see decreases in both glucose consumption and lactate secretion, indicating that reactive oxygen species is promoting the Warburg effect during viral infection. And so you may be thinking, okay, that's cool. What does this have to do with secondary bacterial infections? And we were really excited by this finding because we've previously shown that the secretion of lactate induced by the Warburg effect is contributing to Pseudomonas biofilm growth. And so we showed that using this metabolic inhibitor called sodium oxamate, which is a competitive inhibitor of the lactate dehydrogenase enzyme that is responsible for the conversion of pyruvate into lactate. And so essentially, it blocks the conversion of pyruvate into lactate. And so when we treat interferon-stimulated cells with this sodium oxamate inhibitor, we see a decrease in Pseudomonas biofilm growth, as you can see quantified here. And so we know that the lactate being generated and secreted during the Warburg effect is contributing to Pseudomonas biofilm growth. And now knowing that reactive oxygen species are also contributing to that Warburg effect, we believe that there could be a connection between the production of these reactive oxygen species indirectly contributing to the Pseudomonas biofilm growth that we see. And so I hope at this point you can understand that these metabolic changes induced during viral infections are a really key feature for these Pseudomonas biofilm growth differences that we're seeing. And so in the last few minutes, I want to shift the focus a little bit and talk about some metabolomics research studies that we did, trying to look deeper into this lactate metabolism and also other potential metabolic pathways that are being shifted during viral infection. And so in order to do this, we used this transwell epithelial model. And in this model, we can infect our cells with RSV and then separately collect the cell lysate in order to assess what is happening inside our epithelial cells metabolically during infection, and then apical secretions, which is the environment in which Pseudomonas would be living in the lung. And so overall, during RSV infection, there are a lot of pathways being modified, both inside the cells and in what's being secreted into the apical secretions. And when we look more specifically into the cell lysate, we do see fold change increases in numerous glycolytic and pentose phosphate pathway intermediates, indicating that those metabolic signatures I've been talking about are showing up. And just sort of to tease a little bit where my project is going moving forward with this, when we looked at lactate secretion, we do see an accumulation of lactate in the apical secretions of RSV-infected cells. And surprisingly, we also see these other secondary lactate metabolites called lactol amino acids that are being significantly increased in the apical secretions. And so, well, I don't have time to really dive into these today with you. I do have some preliminary data on how, and we do see that these seem to be inducing Pseudomonas biofilm growth, and that is on my poster. If you'd like to talk to me more about it, I'd be happy to chat more in depth about that with people. Okay, so just to sort of summarize what I've talked about today with you. So during RSV infection, we have our antiviral interferon response that's being induced in infected cells, and this is leading to the production of reactive oxygen species, which is ultimately contributing to this metabolic shift that we see during viral infection that is leading to the production of lactate, and now we know these lactate metabolites, lactol amino acids, that are then being secreted out into the environment and potentially interacting with Pseudomonas to induce biofilm production. And so some of the questions we have moving forward with this project are in these lactol amino acids, looking at when they're being produced, how they're being produced, and when are they, and how are they being secreted from airway epithelial cells. Lactol amino acids have never really been studied in the lung environment or during infection, and so this is a really interesting niche that we're diving into, and there's a lot of questions here for us to sort of pick and choose from on what we want to ask. And we also are really interested in looking at how modulator therapies that many patients with CF are now on could be impacting this metabolic dysfunction that we're seeing during co-infection, as the modulators are affecting so much throughout the lung environment, and so they could be playing a part in changing sort of this environment that we're seeing during viral infections. And on the microbial side of things, one of my big questions right now is trying to look at how these lactol amino acids are interacting with Pseudomonas and what they're doing to induce biofilm. As I said, I have some preliminary data showing that they do seem to be inducing biofilm, we think, through some sort of signaling pathway. And so trying to look at mechanistically what is happening in Pseudomonas that seems to be inducing these biofilm pathways is a major question for me right now. And lastly, more on the metabolic side of things, there are a number of drugs that are used as therapies in other conditions that have this Warburg effect or other metabolic dysfunctions. And so we are very interested in potentially co-opting these drugs as potential therapies to for CF patients during viral infections as a way to try to prevent these secondary Pseudomonas infections and try to improve the overall quality of life for these patients. Okay, so with that, I will go ahead and thank my lab, and I really want to highlight Grace, who is the PhD student working on this project before me and was a major contributor to a lot of what I've talked about with you guys here today. And I will thank our funding, and I'm happy to take any questions.
Thank you so much, Juliana. We have, you're actually a little under. Amazing. So we have about 6 or 7 minutes for questions.
Great talk, Sebastian Riquelme from Colombia. The presence of those lactate amino acids is very interesting. You know that in the world of immunology, it's pretty well accepted now that some metabolites produced during the metabolic reprogramming of immune cells, they regulate the inflammatory function of cells by producing post-translational modifications in proteins that regulate inflammation. So this presence of this lactate amino acid maybe is telling me that maybe this lactate is modifying proteins in the bacteria in the host. Are you going to explore that pathway, that maybe this lactate is producing post-translational modifications in the bacterial protein and maybe that is driving the biofilm phenotype?
Yeah, I mean, that's an interesting idea and an interesting question. The way these lactoamino acids are formed is it's a bond through like the amine group of a lactoamino acid and lactate. And so really it can only seem to happen on like free end lactoamino acids since it needs that amine group for the binding. So there isn't a lot of evidence in literature of it happening on proteins. So it's an interesting idea. Unfortunately, these lactoamino acids have really only been studied for maybe the last decade or so. So there's only like 30, 40 papers on them. So there's a lot of unanswered questions. And that is something we've considered. Hopefully, if anything comes out soon about it being involved in proteins, that'd be something we'd be interested in looking into.
Hi, Dan Wozniak, Ohio State. Very nice talk. I was wondering how you tease apart the potential growth advantage that's being, that's there. So when you see enhanced biofilm aggregates, is it because they're just more bacteria and they're aggregating, or you have the same number of bacteria and there's something in that that's making those existing bacteria aggregate?
Yeah, so I would tend towards the idea that it's something in the environment that is making them aggregate more. Like in vitro, we're infecting with the same amount of Pseudomonas, and we don't see a difference in, say, like attachment really. So it seems to be more something about a signaling pathway or something like that that is inducing these changes. And we've also looked at, like in Pseudomonas specifically for lactate, Pseudomonas mutants that cannot use lactate as a carbon source, and we still see the same growth advantage. And so that indicates it's probably not a carbon source, just like increasing growth, that it's more signaling.
Thanks.
Thank you.
Hi, I'm Jane Jones from Dartmouth. Great talk.
Thank you.
So I was just wondering, I'm not sure about the feasibility of this, but have you ever been able to detect lactate in patient samples, or if you have any mouse models? I was just wondering if that's even feasible.
Yeah, so we do have some like bronchial lavage from patients that eventually we'd like to look at the concentrations. Esteban, who's a new PI at Dartmouth, has just finished setting up a small molecule mass spec, and we're really excited to start looking at samples in this mass spec setup in order to get an actual like concentration.
Thank you.
I have one more question. RSV can demolish cilia. Have you looked at any morphology changes of the epithelial surface or mucus changes, especially when adding Pseudomonas?
Yeah, so in the imaging we are looking at the fact that like the cells are creating a nice monolayer in our experimental setup. We haven't looked specifically to see if there's anything morphologically changing in the cells. The cell line that we use in a lot of our in vitro studies don't really produce very much mucus, so there isn't really too much to look at in that aspect. But
Thank you very much.
thank you.
So now I'd like to introduce our next speaker. That's Erika Orsini. Erika Orsini is an assistant professor of medicine and PhD candidate in molecular medicine at the Cleveland Clinic Lerner College of Medicine. She is currently working in the laboratory of Dr. Rachel Charaga on macrophage mechanosensation, bacterial clearance, and lung injury. Please join me in introducing her, and welcome. Thank you so much.
Good morning, everyone, and thank you so much to the organizers for inviting me to speak. I'm Erica Orsini. I'm from the Cleveland Clinic, and I will be speaking to you today on the role of piezo-1 on CF macrophage bacterial clearance. I have no disclosures. There are many differences in cystic fibrosis macrophages that contribute to chronic lung infection and impaired bacterial clearance. We see reduced toll-like receptor 5 signaling, which is important for recognition and uptake of Pseudomonas aeruginosa. We see alkaline phagocytic vesicles, as well as impaired macrophage phagosome maturation. As you can see, we've highlighted some key genes in the macrophage phagosome maturation pathway, and in key CF macrophage populations, you can see these genes are downregulated. However, we still don't fully understand the cause of macrophage dysfunction in cystic fibrosis, particularly the contribution of the altered CF lung matrix on macrophage function. As everyone in this room is well aware, cystic fibrosis changes the mechanical properties of the lung. We see bronchial dilation and obstruction. We see parenchymal abnormalities such as thickened mucus. There's infection and inflammation in the alveoli, as well as some peribronchial fibrosis. We can measure these changes to the mechanical properties of the lungs clinically using our pulmonary function tests. We see reduced FEV1 as well as reduced FEC. However, how macrophages interact with this altered CF lung matrix for their function remains unknown. Mechanical force, such as that of an altered lung matrix, regulates lung macrophage function. So macrophages in the lung experience a fair amount of mechanical force. They see pressure during tidal breathing and during mechanical ventilation, as well as stiffness from lung injury or fibrosis. This mechanical force is sensed by mechanosensitive ion channels, such as piezo-1 and TRPV4, and their activation in response to mechanical force regulates lung macrophage functions, such as metabolism, cytokine secretion, and bacterial clearance. However, it is still unknown what is the effect of cystic fibrosis on these mechanosensitive macrophage functions. To try to understand this, we looked at some single-cell RNA sequencing data from people with cystic fibrosis and healthy controls, and we saw that in macrophage populations, there was altered expression of mechanosensitive transcription factors. And we think this is important because these mechanosensitive transcription factors regulate downstream mechanosensitive pathways, such as those for mechanosensitive ion channels like piezo-1. Piezo-1 is a ubiquitously expressed mechanosensitive ion channel, and upon its activation by mechanical force, we see an influx of calcium into the cell. And this calcium is an important source for the macrophage for bacterial clearance and actually has been shown to help clear Pseudomonas aeruginosa pneumonia in the acute setting. But we've never looked at macrophage piezo-1 in the CF chronic lung infection. And so we wanted to try to answer this question. And so we posed the hypothesis: chronic Pseudomonas aeruginosa CF lung infection decreases expression of macrophage piezo-1, leading to reduced bacterial clearance in response to the altered CF lung matrix. And to try to test this hypothesis, we examined a few different groups of macrophages. We looked at both wild-type and CFTR mutant bone marrow-derived macrophages. We looked at the R117H mutation, which leads to impaired chloride conductance. And then we also had cystic fibrosis, as well as healthy control peripheral blood mononuclear cells, as well as healthy human alveolar macrophages. We plated them on polyacrylamide gels of pathophysiologic range lung stiffness, 1 kPa being healthy lung, 8 kPa to 25 kPa representing a stiffened or injured lung, and we treated them with and without Pseudomonas aeruginosa flagellin. And we measured piezo-1 expression, as well as calcium channel function in response to the piezo-1 specific agonist YOTA-1, as well as phagosome maturation using pH roto bioparticles. And so what we wanted to look at first was piezo-1 activation in the setting of a CF-like lung environment. And so the two variables we wanted to look at is how does stiffness affect piezo-1 activation, as well as the presence of Pseudomonas aeruginosa flagellin. And so you can see in this first graph that there's very minimal piezo-1 activation in response to YOTA-1 when plated on the 1 kPa or healthy lung stiffness. And you can see there's almost a dose-response relationship where with increasing matrix stiffness, we see further activation of piezo-1, indicating that matrix stiffness is important for piezo-1 activation. And then in our second graph, you can see two dose-response curves. The black is the YOTA-1 alone group, and then the red represents the Pseudomonas aeruginosa flagellin treated group. And you can see in this group, there's a shift of the dose-response curve to the left, indicating that for a given dose of YOTA-1, we see higher piezo-1 activation if the macrophages have been pretreated with flagellin. And so these data indicate to us that in a stiff as well as an environment with Pseudomonas aeruginosa flagellin, we see heightened piezo-1 activation. So next we wanted to see what is the biological relevance to piezo-1 activation. And so we activated macrophages, bone marrow-derived macrophages, using YOTA-1 and treated them with and without flagellin. And we saw that in the flagellin-treated bone marrow-derived macrophages treated with YOTA-1 increased phagolysosome maturation. Similarly, when you downregulate piezo-1 using siRNA, there was decreased flagellin-induced phagolysosome maturation. And finally, to test the relevance of this in the CFTR mutant bone marrow-derived macrophages, we did the same thing, and we were in fact able to augment flagellin-induced phagolysosome maturation using YOTA-1 in the CFTR mutant BMDMs. So finally, we wanted to see how is piezo-1 expression regulated, because we thought this would be important to cystic fibrosis, where we see more of a chronic lung infection rather than an acute infection. And so since piezo-1 function is increased by flagellin, which activates NF-kappa B through toll-like receptor 5, we did a CHIP qPCR experiment where we pulled down the NF-kappa B transcription factor P65, and we did PCR for a piezo-1 enhancer element. And we saw increased binding of P65 to the piezo-1 enhancer element after flagellin stimulation. And no surprise, we see increased piezo-1 mRNA expression when you treat with flagellin. This effect increases the most between 1 to 6 hours and actually starts to wane after 24 hours, which makes us wonder if in the more chronic infection, we actually see reduced piezo-1 expression. And so these data made us interested to see, well, how is piezo-1 expressed in people with cystic fibrosis? And we were able, from our biobank, to get a couple samples of peripheral blood mononuclear cells from people with cystic fibrosis who were getting worked up for transplant. And we found that there was reduced piezo-1 mRNA expression compared to a healthy control. Finally, in order to see the therapeutic relevance of piezo-1 activation, we gave YOTA-1 to cadaveric alveolar macrophages from a healthy donor, and we were able to do something very similar to the bone marrow-derived macrophages. And we were able to increase the flagellin-induced phagolysosome maturation with YOTA-1 in healthy human alveolar macrophages. So in conclusion, cystic fibrosis, chronic lung infection alters the lung mechanical properties as well as expression of mechanosensitive transcription factors. Piezo-1 is a macrophage mechanosensitive ion channel. It is activated in the setting of matrix stiffness and Pseudomonas aeruginosa, and it enhances bacterial clearance upon activation. While piezo-1 transcription increases after Pseudomonas aeruginosa flagellin, this effect wanes after 24 hours. We see reduced piezo-1 mRNA in cystic fibrosis peripheral blood mononuclear cells, and therefore these data suggest to us that reduced piezo-1 expression has a possible role in CF macrophage dysfunction. So for our future directions, we're very interested in working with our collaborators at Case Western to develop a mouse model to study cystic fibrosis chronic lung infection. This has been very challenging because the CFTR mutant mice typically do not get lung disease. So what we've been doing is giving repeated instillation of ACF clinical strain of Pseudomonas embedded in agarose beads to simulate biofilm. We've been giving these infections weekly by bronchoscopy. And I'm just showing some of the histology from one of the mice that we did recently. I believe this mouse got about 5 weeks of infection. And I think we're starting to see some inflammatory cell infiltrate, some consolidation, and hopefully development of some airways disease. So this is ongoing for us to be able to test the therapeutic relevance of piezo-1, as well as many other therapeutics for cystic fibrosis chronic lung infection. So thank you so much. I would like to acknowledge my lab, as well as our collaborators at Case Western Reserve. Their cystic fibrosis research group has been incredibly instrumental to this project. So thank you, and I'll take any questions.
Thank you. It was really nice to see some mechanobiology. Thierry Van Diem from Emory. I was just wondering, which conditions are you really trying to emulate with this? The macrophages are going to be meeting the bacteria in the lumen, right? So do you feel like you're, in the way you've been, using those 8, 1, 8, and 25 kilopascals? Is that
Yes.
what you have as conditions? Do you think that this is sort of relevant to the conditions of the extracellular fluid, the mucus and mucus environment with or without nets, for example, or other things that might increase the stiffness? Or are you mostly interested in looking at mesenchymal infection, which is really not happening that much in CF?
I think what we're trying to recreate is the stiffness of like an alveolus filled with fluid and infection,
Okay.
Because that becomes dense and more stiff. I agree. There's opportunity to study other different types of mechanical forces that may have more relevance to cystic fibrosis. So I think this is just the beginning. We're using stiffness as a way to activate our channel, but I think there's definitely opportunity to try other things such as flow and changes in pressure and other things, because obviously the cystic fibrosis lung matrix is different, and I think we need to find a way to copy that in vitro to study it more.
So specifically, do your conditions, you think, mimic some of the changes in the stiffness of the extracellular microenvironment, the mucins themselves? Have you sort of looked at the stiffness of the extracellular matrix, the one in the lumen?
Oh, of like mucus impacted lumen?
Yes.
I have not yet, but we do potentially have the opportunity to get some explanted lungs, although obviously there aren't as many transplants anymore, which is a great thing. So maybe there's a potential opportunity to do atomic force microscopy on these different lung sections, so we can be a lot more precise in vitro.
awesome.
I
Thank
agree.
you.
Thank you.
Thank you, Erica. If I could ask another question, I was interested in your finding that piezo-1 mRNA is decreased in macrophages from people with cystic fibrosis. I was wondering about the mechanism behind that. If you could comment on, how, if there's an upstream link between CFTR dysfunction and piezo-1 loss of mRNA, or if that's, does that just reflect interactions with Pseudomonas, as your data might suggest, or is there some other connection between the two?
I think there probably is another connection, because we have looked at the CFTR mutant BFDMs from the mice that do not have lung disease, and we actually have seen some changes in piezo-1 expression and activity. We haven't yet understood the mechanism behind that, but it does bring into question whether or not there's also channel-channel interactions, and calcium homeostasis is very altered in cells that have CFTR mutation. So I think for this particular project, we're focusing on kind of exhaustion of NF-kappa B, chronic inflammation, chronic infection, and having piezo-1 be downstream of NF-kappa B, and then having reduced expression in that setting. But I agree, there's probably much more complexity to the piezo-1 CFTR story.
Interesting. Now, there's so much, that we're learning about connections between ion channels. Also begs the question as to what's happening to piezo-1 in the epithelium or other lung cells.
Absolutely.
Tremendous. Any other questions?
It was a really exciting talk. Thinking about stiffness is something I think with macrophages we need to think about more. The use of flagellin is really interesting to me, because there's data that, although there's a lot of heterogeneity in the Pseudomonas population in the lung, that Pseudomonas often loses flagella when it becomes chronic. And I was wondering if you're going to have the opportunity to look at like early, late isolates to be able to see if you see differential effects.
That's a very fair question. What we have found, because we've also worked with LPS, is that this is not specific to flagellin. It's actually just specific to NF-kappa B activation. But that is a very fair point. that we do see decreased expression of flagellin. However, we do continue to see some flagellin. There are papers out there that actually measure flagellin concentration even in very late stage cystic fibrosis. So that's how I also justify using flagellin, but it's actually not flagellin specific effect.
I think we'll take one more question.
Hi, Ben Kopp, Emory. Really nice talk. When in your data mining or in your pathway analysis with this, have you looked at the connection to ROBO1 and SLIT and that axis, like the ROBO-SLIT axis with piezo1? Does that control some of the other mechanosensitive forces?
I have not. I'm not familiar with it, but I'll definitely look into it.
Okay, because it's pretty active like in the cancer biology of this, so it might be relevant here too.
Okay, absolutely. Thank you.
Now I'd like to introduce Cheryl Fernandez. Cheryl Fernandez is a postdoctoral scientist in the laboratory of Dr. Peter Chort at Cedars-Sinai Medical Center in Los Angeles. She's investigating mechanisms of enhanced virulence that are selected or evolve under chronic antibiotic exposure in the CF pathogen Pseudomonas aeruginosa.
So many in the audience must have heard of talks where they discuss about antibiotic efflux pumps with respect to antimicrobial resistance. But today I'm going to show you data which demonstrates how inactivation of the MEX-EF-OPRN antibiotic efflux pump can enhance the virulence of CF Pseudomonas isolates. I have nothing to disclose. So Pseudomonas infections are very common in persons with CF, but in about 2019-2020, we saw a decline in the prevalence of Pseudomonas infections, and this could be attributed to the approval of CFTR modulators for therapy. So as more and more persons with CF begin modulator therapy, it becomes important for us to understand how CFTR modulators can actually influence CF infections. And so therefore, these two studies looked at two independent CF cohorts, and they concluded that while the Pseudomonas bacterial burden decreased in the lungs, the infections persisted even months after ETI treatment. So it is important for us to study how chronic modulator as well as chronic antibiotic therapy can influence the evolutionary landscape of Pseudomonas, particularly from the perspective of infections. And in this talk, I will be touching upon the second question of how chronic antibiotic therapy can result in augmentation of PA virulence mechanisms. So aztreonam is a beta-lactam antibiotic that is commonly prescribed to treat Pseudomonas infections in CF. And in this study, it shows that persons infected with aztreonam-resistant PA isolates were more likely to be hospitalized and suffer from pulmonary exacerbations compared to individuals who were infected with aztreonam-sensitive PA isolates. Around the same time, my postdoc mentor, Dr. Peter Jorth, he was pursuing his postdoctoral studies in Dr. Pradeep Singh's lab, and he was curious about the clinically relevant mutations that evolved in aztreonam-resistant Pseudomonas populations. And so what he did was he did a genome analysis of in vitro evolved aztreonam-resistant PA populations, and he found that there were several mutations in the genomes of these populations, and some of these were inactivating mutations in the MEX-EF-OPRN operon that encodes the MEX-EF-OPRN antibiotic efflux pump that effluxes quinolones and chloramphenicol. Later, my colleagues showed that the absence of this efflux pump was sufficient to increase lethality in infected mice. And so at this point, we had two questions. Is this increased lethality because of exacerbated pro-inflammatory responses, or is this because of increased bacterial virulence? And so what I did was to repeat these acute infections in wild-type mice, but to sacrifice them at an earlier time point and to quantify the lung bacterial burden. So we found that the mutant-infected mice had higher lung bacterial burdens, and they also showed increased lung barrier damage, which is indicated by the presence of bacteria at secondary sites of infection. Complementation of this knockout significantly reduced bacterial lung burdens and also decreased lung barrier damage. So then we wanted to identify what are the factors which could contribute to the increased virulence of the mutant. And we found that there were several virulence genes that were upregulated in the mutant compared to the wild type. But we chose to focus on these two genes, that is LASB and RHLA. So RHLA is a gene that is involved in the biosynthesis of rhamnolipids, which is a biosurfactant that can be found in the sputum of CF individuals infected with Pseudomonas. And there are studies which show that strains that produce high amounts of rhamnolipid cause increased nasal epithelial damage compared to strains that produce low levels of rhamnolipid. And LASB is a gene which is involved in elastase biosynthesis, and like rhamnolipids can cause airway barrier dysfunction and hyperinflammation. So to test the roles of these two virulence factors, we made knockouts of LASB and RHLA in the mutant background. And we observed that while LASB was also involved in the colonization of the lung, we found that abrogating either LASB or RHLA expression reduced lung barrier damage. So now all this data has been accumulated from acute infections in wild-type mice, but what about CF models? So we did chronic infections in the beta-ENaC mice, and so these mice overexpress the sodium ion channels in the lung and the airway, resulting in mucus accumulation, which is characteristic to CF lung disease. And we intended to enumerate the bacterial lung burdens at day 7 and even neutrophils, which would give us an idea of the inflammatory status of the lungs. However, by day 4 itself, we found that 50% of the mutant infected mice died because of the infections. And at day 7, if we compared the lung bacterial burden in the mice that survived, there was no difference between the wild type and the mutant infected groups. However, the mutant infected mice showed high number of neutrophils, indicating that there was more inflammation in the lungs. Further, we wanted to test the roles of elastase and rhamnolipids. So for this, what we did was to establish air-liquid interface cultures of differentiated CF airway epithelia, and we exposed these epithelia to supernatant from either wild type, the mutant, or mutant not expressing elastase or rhamnolipid. So the supernatant would contain these secreted virulence factors, elastase and rhamnolipids, and a decrease in the transepithelial resistance below 330 ohm centimeter square was considered as CF airway epithelial barrier damage. So we observed that the epithelial barriers only exposed to the mutant showed a decrease in transepithelial resistance, and abrogating elastase and rhamnolipid expression reduced CF airway epithelial barrier damage. Next, we wanted to identify how prevalent is this mutation in CF isolates. For this, we analyzed the genomes of 186 CF isolates and found that 53% of the isolates had non-synonymous mutations in either MEX-E, MEX-F, or OPRN genes. Additionally, we found that 21 of these isolates had inactivating frameshift or nonsense mutations in either MEX-E or MEX-F, indicating that the efflux pump was dysfunctional in these strains. We chose to further focus on the clinical isolate, that is the Liverpool epidemic strain, LESB58, because LESB58 is highly transmissible and it is one of the first CF epidemic strains to have been identified. It also results in pulmonary and extrapulmonary exacerbations, and the mechanisms of its enhanced virulence are unknown. So we thought that the inactivation of the MEX-EF OPRN efflux pump was probably resulting in the virulence of LESB58. To test this out, we complemented LESB58 with the wild-type MEX-EF OPRN operon, and we first quantified the expression of the two virulence factors that we found were important, that is LASB and RHLA, and we found that complementation significantly reduced expression of these two virulence genes. Coherent with these findings, we found that complementation also abrogated CF airway damage, indicating that the hypervirulence could be possibly because of the MEX-EF OPRN mutation. So in conclusion, we see that inactivating mutations in MEX-EF OPRN are prevalent in CF clinical isolates, and these could enhance the virulence of these Pseudomonas isolates. And such inactivating mutations may be selected by chronic beta-lactam exposure. In fact, in support of our findings, a recent clinical study was published wherein authors followed 5 patients who were chronically infected with Pseudomonas and were receiving beta-lactam therapy. 4 out of these 5 patients had isolates that evolved inactivating mutations in MEX-EF OPRN, that is indicated by these black circles. More importantly, 3 out of these 4 patients soon expired after the identification of these isolates. So there seems to be a potential clinical link between deleterious mutations in MEX-EF-OPRN, chronic beta-lactam therapy, and patient mortality, which is all the more reason for us to test this mutation as a contributing virulence promoting factor in the clinical isolate LESB58. And so we plan to do chronic infections in beta-ENaC mice. We plan to quantify the lung bacterial burdens and look at the lung function as well as lung inflammation. We also have some patient data for the clinical isolates whose genomes we have analyzed. And so we want to see if there is any correlation between the lung function and MEX-EF-OPRN mutants in these patients. Finally, I'd like to thank my postdoc mentor, Dr. Jorth Lab, my mentoring committee, especially Dr. Heisert and Dr. Bergina for their clinical perspective, collaborators from the Southern California CFF chapter and UT Austin, my friends in the Cystic Fibrosis Trainee Network. I have a poster presentation tomorrow. It's poster number 74. If any of you want to discuss about how the deletion of an antibiotic efflux pump is actually regulating the expression of virulence factors, you can meet me there. I would also like to draw the attention of CF trainees in the audience. We have a very vibrant CF trainee network, which meets once a month, and we discuss our research, we exchange resources, and we discuss transitions to careers in academia and research. So if you'd like to join us, you could scan this QR code, or you could contact me on my email or meet me at my poster, and I can help you with it. Thank you.
Yeah, thank you very much. Very nice talk. I was interested in your in vivo data where you showed that your mutant in the efflux pump is basically, if I understood right, killing mice through inflammation, and you showed neutrophil, the presence of neutrophils. Did you have a chance to look mechanistically what do these neutrophils do? Are they, because the presence of neutrophil is not necessarily a proof of damage, and so have you looked at whether they're activated, whether you have got lung damage, etc., or maybe it's within your plans that you...
Yeah, so we are planning to look at the inflammation and the cytokines, because also recruitment of neutrophils means more cytokines. So maybe that could be one of the reasons why we are seeing increased death. Yeah, and LASB also has been shown to inactivate a lot of cytokines and complement, etc. So I wonder whether that's a link with your neutrophil data. Yes.
Really nice talk. Thank you. 2 questions. One is, it's very scary that resistance mutations also causes hypervirulence. Have you identified any conditions where the MEX-EF mutations produce a fitness cost for the bacteria?
MEX-F mutations, no, I have not looked at fitness costs, but there are others who have looked at whether the mutations result in a fitness cost, even overexpression, or in my case, the inactivation does not cause a fitness cost in vitro growth conditions, at least.
And not in you have no animal competition experiments where you...
Pardon me?
Have you done any animal competition experiments where you put a wild type?
No, I haven't.
That's cool. The other question is the finding of the MEX-EF mutations in the Liverpool epidemic strain. Is that a general phenomenon of the Liverpool epidemic strain background, or was that a particular variant of LES that contain that mutation?
I think it's in the background because a lot of the strains derive from the LESB58 lineage. They all have the MEX-F inactivating mutation.
Thank you.
Oh, great talk. You said over 50% had inactivating mutations in the efflux pump. I was curious, did you look at mutations in MEX-T, which is the transcription factor that controls expression of the...
Yeah, so a lot of people have looked at MEX-T mutations, but since it's a transcription factor that can regulate a lot of other things apart from just the efflux pump, we have kind of recreated the mutation only in the efflux pump because our question is whether the efflux pump is enhancing virulence.
Thanks for a really interesting talk. I was curious if you had looked at the expression levels of any of the other efflux pumps in that mutant. Is there a compensatory upregulation of one of the other 12 R&D efflux pumps?
We do see a slight increase in MEX-GH, but the MEX-GH, there isn't much known about whether that really affects the virulence. But yes, that's a good question. Only MEX-GH was slightly upregulated, but the other efflux pumps, they had no difference in expression.
Thank you.
Not a problem.
Thank you very much, Cheryl. Up next, I would like to welcome Sohail Nevis. He's an assistant professor in the Department of Biochemistry, Emory University School of Medicine. His lab utilizes RNA, protein biochemistry, yeast genetics, structural biology, and cell-based assays to understand post-transcriptional regulation of gene expression in human fungal pathogens and applies this knowledge to identify novel targets for therapeutic development. Welcome.
Thank you. And thanks everyone for coming here. We have nothing to disclose. So people with cystic fibrosis have this thick mucus, which is hard to be cleared and therefore provides a very good environment for different pathogens to thrive. And we heard a lot of nice information about the relevance of viral and bacterial pathogens in this context. And I'm going to switch gears here and talk about these other pathogens in CF, which are fungal pathogens. So CF patients are commonly colonized or infected with fungal organisms, and depending on the literature you look at, that can range from 5 to 78%. I would go more towards the bigger numbers. The most prevalent fungal organism here is Candida albicans, which is associated with significant increased rates of hospital-treated infections and also reduced FEV1 decline. It's also the persistent colonization of Candida is associated with severe cases of pulmonary infection. And to make matters worse, Candida is often found in mixtures with different organisms, different pathogens, especially Pseudomonas aeruginosa, which is then contributes to the exacerbation of the infection. So let me tell you a little bit about Candida. Candida is an opportunistic fungal pathogen. It's harmless to healthy patients or healthy people. But in immunocompromised patients, it can be really devastating. It targets multiple organs. Anywhere there is epithelial, you find Candida, including lung. And the death toll from Candida infection each year is about 400,000 to 500,000 people, which is a crazy high number. And the problem is the resistance to antifungal drugs is on the rise, mostly due to global warming. Candida exists in two morphological forms, yeast and hyphae. So normally it's found in an environment is in the yeast form, but once it feels that it's in the human body, things like 37 degrees CO2, Glocknax serum, anything, it would switch to the filamentous form. And this yeast to hyphae transition is very important for the pathogenicity of Candida. In this graph here, this is the skin equivalent. I honestly don't know how they make it. But if you use if you put Candida on top of that, within a couple of hours, they form hyphae and destroy the tissue. They'll penetrate deep into the tissue. But if you use mutants which cannot form hyphae, there is not much of damage caused, maybe some inflammation, but that's about it. So this yeast to hyphae transition is very important for the pathogenicity. And it's a very complex process. It's controlled by many transcription factors, a lot of RNA transcriptomic changes happening. Some of these transcription factors or programs are pro-yeast, so they favor the yeast form. Some are pro-hyphae, they favor the hyphae form. And the yeast transcription system is actively suppressing the hyphae formation. So when they sense that they have to go from yeast to hyphae, these pro-yeast factors should go away. So their mRNA should be targeted and degraded. And that's what my lab studies, which are like what regulatory mechanisms control recognition and destruction of those pro-yeast mRNAs. So whenever we talk about recognition of mRNA, it's usually RNA modification involved. In eukaryotic mRNA, we have over 100 different modifications to the mRNA. The most prevalent internal modification is M6A, which is the deposition of a methyl group here. This doesn't happen randomly. It's a sequence context, and there are these writers or metyl transferases, methyl 314 in human, IME4 in yeast. And you can imagine these methylated RNAs, they're more hydrophobic, so their biophysical properties changes, which then results in downstream effects such as changes in splicing, RNA export, RNA decay, and so on. So the best studied cases of M6A are in mRNA decay. So that's why we asked the question whether M6A plays a role in mRNA decay during yeast to hypha transition in Candida. So we purified RNA from different stages of hypha formation in Candida and looked at the amount of M6A using an anti-M6A antibody compared to the total RNA. And we see that indeed during hypha formation, there's a transient deposition of M6A on these mRNAs, which then go away. We identified the methyl transfer is responsible for this called IME4 and genomically tagged it with a MIG tag, and we showed that its expression is also nicely correlates with this deposition. So what happens if we remove that methyl transferase from the genome of Candida? We end up with shorter hyphae, as you can see here. The addition to the surfaces, plastic surface, which is the early stages of biofilm formation, goes down dramatically. And they also cannot bind, as well as the wild-type Candida, to different human epithelial cells. So it's a good target, right? So IME4 is an enzyme. Any enzyme can be targeted by small molecules. Unfortunately, the human homologue of IME4 methyl-3 is the target for many cancer drugs, which are at different stages of development. So we bought the ones that we could buy here on this list and tested their effect on Candida. Not much of effect, which we can discuss later why that is. Except for sinafungin, which showed some promising results. First of all, it decreased hypha formation greatly, as you can see here, even at very low concentrations. And so these are like short-term biofilm hypha formation, but even also in the long term on this spider media, you kind of look at hypha formation in 5-6 days. This hairy, creepy-looking colony is like without drug, but then if you use sinafungin, there is really no hypha formation, even within 5 days. It also very effectively reduced the addition of Candida in short time to plastic surfaces, and also if you preform these like biofilms and then apply the drug, we can see that the level of biofilm goes down within like 24 hours. It also reduced the addition of Candida to different human epithelial cells. So at this point, we wanted to see how it works in like animal models. So we use these models called Galleria melonella or waxworm. They are fishing baits. You buy them from fishing companies. But despite their look, they're really respected model organism in infection studies. They have very complex innate immunity. You inject pathogen to them. If pathogen is bad, they turn black and they die, but otherwise they remain healthy. So we looked at the effect of sinafungin in pathogenicity of Candida in these worms. If you don't add sinafungin, just Candida, within 5 days, all the worms die. But if you add sinafungin, you can see a very well rescue. And even so, this is not the highest concentration. It's actually not the lowest and not the highest, so kind of in between this magic number of 0.15 milligram per kilogram of the organism. And sinafungin by itself has no bad effect on these worms. So it's not toxic to the worms a lot, so which is a good thing. So at this point, we wanted to look at a more practical context. So we teamed up with Niall McCarthy lab at Emory to look whether this mRNA methylation in Candida contributes to the pathogenicity of Candida in CF models, and whether it can be targeted by sinafungin. So first, we looked at the addition of Candida to cell lines of wild type or CF cell lines, and we see that compared to the wild type, the M6A deficient Candida cannot adhere to these cell lines very well, both wild type and CF. An interesting observation was that Candida, wild type Candida, binds better to the CF cell line than the wild type, and that's something we are looking into at the moment. Then we looked at the addition, so whether sinafungin does the same thing here. And so we looked at the effect of sinafungin on addition of wild type Candida to different CF cell lines, wild type or CF cell lines. And we see a nice dose-dependent response. More importantly, sinafungin has no bad effect on those cell lines. So we looked at the proliferation of these cells or the toxicity using lactate dehydrogenase release in the media. And we see that at these low concentrations, there is really no bad effect from sinafungin. So everything so far has been done with SC5314, which is a standard strain of Candida. So then CF Virus Repository Center at Emory was kind enough to isolate Candida species from sputum of 3 patients, which we call 233, 488, and 833. And we see that sinafungin has the same effect on those. So those guys bind to wild type or CF cell lines, and when you add sinafungin, that adhesion goes down dramatically. So as I told you, Candida and Pseudomonas play together in CF context, right? So we wanted to see what is the contribution of fungal mRNA methylation to this interaction. So we used a PA01 strain which expresses a GFP molecule. So anything which is green here is Pseudomonas. So we just looked at the addition of Pseudomonas to the surfaces here in the presence of Candida. So PA01 by itself binds that much. Once you add Candida, wild-type Candida, that binding goes up a lot, which has been reported in the literature, so nothing new. But once we use an M6A deficient mutant of Candida, that addition goes down again. So next question is whether sinafungin has the same effect. So what we did, we again use this fluorescently labeled PA01. As you can see here, PA01 by itself binds that much to the surface. After 2 hours, when you add Candida without sinafungin, that addition goes up a lot. But even a tad bit of sinafungin you add to the environment, and that addition goes down dramatically. In the Galleria organism, if you mix Candida and Pseudomonas together, instead of 5 days, in 2 days, everything is dead, as you can see here. But then again, you add sinafungin, and you see that up to 80% of the worms can survive. And last, we show that Pseudomonas can bind better to human cells in the presence of Candida, comparing like this one to this one. But again, as you add sinafungin, that adhesion goes down. So last, ETI has been shown to be an antifungal in certain fungi, for example, Aspergillus. So you add ETI to the media, and Aspergillus biofilm formation, growth, and everything goes down. We wanted to see whether it has the same effect on Candida. We did not see any effect on the growth or adhesion of Candida to the surfaces. Different combination of these 2 or all 3 together, no effect. And also, if we treat cells before addition of Candida with or without ETI, and then add Candida, we don't really see much of a difference between addition of Candida to these cells. So in conclusion, we showed that the mRNA methylation is critical for the pathogenicity of Candida in human airway epithelial cells, and sinafungin can reduce this addition. So it's a very promising small molecule to treat fungal pathogens. And also that the interaction of Candida with Pseudomonas depends on the fungal mRNA methylation and can be targeted by sinafungin. So what we want to do in future is, first of all, determine what is the mechanism here. So what mRNAs in Candida are targeted by M6A modification, and we are using nanopore technology to address that question right now. Another thing is to look at the effect of sinafungin. So everything we have done so far was cell lines, right? So, but we want to see what the effect of sinafungin or fungal mRNA methylation is in the context of primary cells. First of all, like, is there any preference for any kind of cell in there? And also establish all these assays in a mouse model. And last, we are starting to look into the response of the epithelial cells into fungal infection. By that, I would like to thank my lab, especially Anushka and a very talented undergrad in my lab, and Azam, a postdoc in my lab, who have contributed a lot to this work. And also my 7-year-old son, who enthusiastically counted live and dead worms for several weeks. And I am grateful to the CF research community at Emory, especially McCarty Lab, Niall, Going, Analia, and Camry. They have been very helpful, and CF Discovery Corps, Arlene, and Katie for providing us with the cells, and our fundings, NIH and Emory, and thank you for your attention.
Hi, Katie Heisert from National Jewish Health. That was so interesting. And I started your talk thinking, as a clinician, we never treat Candida. Why do we care about this? We don't think it's an infection. And then you got to the Pseudomonas data. And so I think all of your experiments was simultaneous Candida and Pseudomonas. Have you done any work seeing if you establish the Pseudomonas first and add the Candida, do you get enhanced? Do you think we should be treating Candida in our patients to detach the Pseudomonas?
We have not done that experiment, but we would love to do that. So that's on our to-do list. So we are actively working on that. And yeah, definitely we want to do that. As to whether you have to treat Candida, I would say yes, but I'm not a clinician and I don't have the data for that.
Thank you for the talk, Thierry Van Zien from Emory. So I think we'll take some of the questions back home as well. But first of all, thank you. I think it's beautiful work. We know that the immune system is really the interface between fungi and mucosae. So there's a lot of macrophage, Candida interaction, neutrophil, Candida interaction, patients with immunodeficiencies, fungal infections bloom. And there's recent literature, especially on MET-L3 as a M6A promoting, enzyme in macrophages and neutrophils. It's been shown to be actually critical to their function in mucosae. Do you know if sinefungin has any effect on M6A modifications in myeloid cells?
So sinefungin, but it should have, but again, honestly, we haven't tested that. So at concentrations that it hits Candida, it has no effect in human, which can have many reasons. I don't know, really, maybe Candida has a special transporter for it, which can suck it up from the environment way better than human cells, for example, because it's a pan-methyltransferase inhibitor, so it should target anything. So I assume at higher concentration probably has effect on M6A methylation among other methylations in human cells, but then at those concentrations you might start seeing toxicity, right? But again, the methylation pattern of hosts in response to fungi has not been studied. We know it changes in response to viral infections, for example, very well. So those studies have done beautifully. So that's one thing that we are trying to establish. So it's like basically next.
Let's work on this together.
Yeah,
I think
sure,
we can provide the lung conditioned immune cells for you. So we know there's a lot of transcriptional changes when they get into the lung and then they get exposed to the pathogen cues. So I think we can easily work on that together. Thank you.
So Hale, a related question. That was tremendous. Thank you. I know that was a little weird, right? I'm like over here. I was wondering if you could comment a little bit on the yeast to hyphae transition in the lung. For example, if you take a person with CF and they acquire or inhale Candida albicans in its yeast form, is there just kind of a basal level of yeast to hyphae transition, or do you think that that's induced by some condition? And I think of it specifically at the beginning of your talk when you cited the data showing that presence of Candida is associated with pulmonary exacerbation. So, do you think the yeast to hyphae transition happens kind of all the time slowly, or do you think it, happens and then it's followed by an exacerbation, or how do we think about this in a clinical context?
That is not clear for several reasons. First of all is that, so once they isolate Candida from patients, it's already like many steps after the infection has happened, so you take it to the lab, culture it and all that, and then they can transition back if they're in hyphae to yeast form and so on. So you can't really tell. The best way to answer that question is to get samples from the lung, infected lung, and then do an RT. There are genes which are specific to yeast, genes which are specific to hyphae. So then you look at by using RT to see in which state these cells are in the lung. right? Not that you extract them, take them to the lab and study, right? Which that's something we are going to do with our mice studies, right? Another thing is that yeast is usually associated with dissemination. So they exist in our body. So first of all, Candida is not airborne, right? So unlike Aspergillus. So that's why probably when people talk about the fungal infection, they mostly talk about Aspergillus because it's airborne and all that. But Candida, we have Candida in our body all the time, right? So one thought is that they're mostly in the yeast form, so they're kind of hiding and so on. But then once the situation is good, like immune system goes down or whatever, they transition to hyphae and they start the damage. One thing to note is that several groups have isolated Candida from CF people, right, in large numbers. And what they have noticed is that frequently you see mutations in different genes which are associated with hyphae, as if all those which have been isolated from Candida people, from CF people, they are kind of forced to be in hyphae form, right? Like the transcription factors which prefer hyphae form and things like that. So that is not quite clear really in the context of infection. I think they normally kind of hide in the yeast form, but then once the situation is good, maybe you have, I don't know, Pseudomonas infection or whatever, then they go into hyphae form and start the damage.
Cool. Thank you. Much more to learn there. Any other questions? None? Okay, great. Thank you, Sohail. So I'd like to introduce our last speaker then, Sebastian Riquelme. Sebastian is a biochemist, immunologist, and microbiologist who works at Columbia University as an assistant professor, where his laboratory studies how metabolism regulates the outcome of infectious diseases in the human lung. Please join me in welcoming Sebastian.
Well, first of all, thanks very much, chairs, for inviting me. And thanks, Maria, for you for sticking until the end. So the story that I will tell you today, well, disclosure, no disclosures. So the story that I will be telling you today, it's about, it started with a collaboration with a friend of mine, Dr. Clemente Brito from Yale University, in where he shared with us some sputum samples. And normally in my laboratory, what we do metabolomics to everything. If you go to my lab, we will do metabolomics on you. So, and we decided to do metabolomics analysis in these samples. And the samples, the characteristic of most of the samples from the sputum samples from these people with cystic fibrosis, that most of them they were infected with Pseudomonas aureus. So we wanted to do a correlation what happens between the presence of these Pseudomonas and which metabolites are accumulated, and what is the role of that metabolic response, that metabolic environment. And after doing a very exhaustive analysis and bioanalysis, what we found is that there were particular metabolites that were enriched in the airway of people with cystic fibrosis. And these metabolites were these ketone bodies, such as beta-hydroxybutyrate, as you can see here. And there are different types of ketone bodies, but one of the most important is beta-hydroxybutyrate. And this is telling us that these metabolites are associated with a process that is called ketogenesis. Okay? Maybe some of you already have learned about ketogenesis. Maybe some of you are already on ketogenic diets, trying to lose weight. Very good. So we will be talking about ketogenesis today, okay? So what is ketogenesis for the people in the audience that are not familiarized with this process? So, for example, if you didn't have breakfast today, as I did, which is a very bad idea, so your organs will be under a bioenergetic crisis, okay? And this can compromise the integrity of your organs. And your mitochondria won't be happy. So what will happen is that your cells will start suffering from this bioenergetic stress, and if your tissue don't regulate this bioenergetic crisis, they will promote tissue necrosis and will lead to inflammation, and this, at the end of the day, will lead to organ damage. But because we have a very good system, our body is very well prepared for all of that. There is a very important organ in our body that is called the liver, and this liver, what it does is that it takes up some fatty acids from the bloodstream, or also communicate with adipose tissue to capture these fatty acids. And through a process that is called fatty acid oxidation, activates a process that is called ketogenesis. And this ketogenesis synthesize and release into the bloodstream these ketone bodies, such as I told you before, this beta-hydroxybutyrate. And these ketone bodies, they travel to this damaged tissue and they fuel the mitochondria and they help the mitochondria to produce energy. And this mitigates the bioenergetic crisis and the inflammation occurring in the tissues. So after that introduction of ketogenesis, I think you already got the idea that ketogenesis will be anti-inflammatory during this process, okay? But our question is, what is the role of ketogenesis during Pseudomonas pneumonia? Why are those ketone bodies being accumulated in the airway of people with cystic fibrosis? What is their role during the infection with Pseudomonas? So to answer that question, what we did a nutritional intervention, which is something that maybe you are doing on yourself, where you are consuming ketogenic diets. And what we did is that we take two groups of animals and we fed them for 8 days at Libitum with either a controlled diet in where almost all of the calories are derived from carbohydrates, or we expose them, the other group, to a ketogenic diet in where mostly all of the calories are derived from fatty acids. As you can see, we maintain constant amount of proteins. We didn't see any changes in body weights during these short periods. So if you have that question, that is solved. And the way we did is that we exposed these animals to a collection of different Pseudomonas aeruginosa isolates derived from the airway of people with cystic fibrosis, just to understand what is the interaction with this ketogenic environment. And then we analyze how the ketogenesis program in the host altered or impacted the airway inflammatory response to the pathogen. As you can see here in the airway, animals infected, control animals infected with Pseudomonas aeruginosa, they already activate ketogenesis in the airway and they accumulate this ketone body beta-hydroxybutyrate, which was increased even more in animals that were under ketogenesis. So this is demonstrating us that the ketogenic diets, they were really working on promoting this ketogenic environment. And something super important is that the pathogen burden didn't change during this ketogenic environment. So everything that I will tell you now has nothing to do with the pathogen burden, but instead is only associated with immunoregulation. Clemente just arrived. I cite you. Yeah. So as I told you before, the ketogenesis was associated with an anti-inflammatory response, and we were expecting this. And in fact, this is what happens, because if you analyze the concentration in the airway of different tissue-damaging cytokines, such as IL-6 or TNF-alpha or IL-12P70, all these cytokines were decreased in the host under ketogenesis. And other cytokines that participate in the recruitment of neutrophils, which we know are very important in cystic fibrosis, they were also suppressed in the ketogenic environment. And of course, the amount of neutrophils that were infiltrating the airway during infection decreasing at 50%. And all of this is telling us that this metabolic program coordinated by the liver has nothing to do with the pathogen burden, but instead suppresses inflammation. And this is something that in immunology we know as a mechanism of defense that is called disease tolerance, in where the host, to try to limit the immunopathology triggered by the infection, what it does is that it suppresses the inflammatory response. And the pathogen take advantage of that immune suppression, and because the host is not trying to fight the infection, but instead is trying to preserve the organ integrity, and the host and the pathogen, they come into disagreement, and the pathogen exploits that environment, and now the host and the pathogen, they coexist. And this is what we call the host-pathogen coexistence, or clinically, it's an indolent infection or a chronic infection. So said that, then we wanted to understand, okay, if these ketone bodies are there in the airway of people with cystic fibrosis, and Pseudomonas is evolving in that environment, maybe the Pseudomonas strains that we are collecting from the airway of people with cystic fibrosis, they have learned how to trick the system and how to exploit this ketogenic environment for their own benefit. So to analyze that, to study if Pseudomonas from the airway of people with cystic fibrosis were triggering ketogenesis in the airway, what we did is that we took animals and we challenged these animals with a collection of 17 strains together from a chronically infected patient with cystic fibrosis, which is a tolerant patient, and we compare the immunopathology triggered by these strains with the disease promoted by wild-type PO1, which is the classical laboratory strains of Pseudomonas aeruginosa, which we know that is very, very inflammatory, highly pathogenic, and that kill the host very quickly. As expected, we found that this clinical isolate of Pseudomonas aeruginosa, they induce a much lower immunopathology in the airway because they didn't trigger the accumulation of, or they triggered much less accumulation of IL-1 beta, IL-6, and TNF-alpha, which we know that are cytokines secreted by the immune cells and promote tissue damage. At the same time, these Pseudomonas isolate, they didn't kill the host in comparison with PO1, meaning that they are able to preserve the integrity of the host. And maybe you're wondering, or you will be thinking that maybe the host, they survived to the infection by these Pseudomonas isolate because the host already cleared the infection. But that is not the case, because when we analyze the bacterial burden, you can see that the bacteria are there. So they are coexisting with the host. They are exploiting that they are adapted to induce this disease tolerance mechanism. So Pseudomonas aeruginosa evolves in the airway of people with cystic fibrosis to exploit this immune mechanism to preserve the airway integrity, and that is associated with ketogenesis. Now, the question is, how this Pseudomonas aeruginosa promotes ketogenesis in the airway and the accumulation of ketone bodies? And this is one of the questions that is keeping me up during the night. To answer that question, what we did is that we we compare how, if these clinical isolates were inducing the same type of ketogenic response that this laboratory strain PO1, which everyone uses. And we found that the accumulation of beta-hydroxybutyrate in the airway was exactly the same between the two types of pathogens, either these highly inflammatory strains or the clinical isolate that we know that exploit disease tolerance. And as I introduced you before, ketone bodies are normally produced by the liver, and we can track hepatic ketogenesis by measuring the same metabolite but in blood. And we were expecting that maybe the level of ketone bodies in blood was going to be the same. But when we did the experiment, we found that the clinical isolate, they didn't induce much of a hepatic ketogenic response in comparison with PO1. Suggesting that maybe they are exploring another different mechanism in the airway to enrich the local environment with ketone bodies. And this is completely independent of the liver. Thinking about that, we decided to interrogate the airway metabolome and to ask the question, so if these ketone bodies are not coming from the liver, from where are they coming? Maybe there is a local source of ketone bodies that this bacteria, they learn how to exploit. And we were looking for that. So first of all, we know that ketogenesis is regulated at the metabolic level. So for that, what we did is that we analyzed the airway metabolome of the animals infected with these clinical isolates. And what we found is that in addition to cluster as the most concentrated metabolites in the airway of these animals, these ketone bodies, beta-hydroxybutyrate and acetoacetate, these metabolites cluster with itaconate. And maybe you already hear about this metabolite. It's a very fancy metabolite that is produced by the mitochondria of myeloid cells. And the role of this metabolite is that it's anti-inflammatory, it's immunoregulatory. So maybe these pseudomonas, they have evolved in the airway of people with cystic fibrosis to enrich the local environment with ketone bodies by exploiting the production of itaconate by the host. And something that I didn't tell you before, itaconate is produced by the mitochondria, which is super important here. So maybe there is an axis, a platform between itaconate, a cross-link between itaconate and ketone bodies, and pseudomonas have learned how to exploit this metabolic pathway in the airway. So to evaluate the hypothesis that it's really itaconate which is promoting this ketogenic environment, what we did is that we took animals that can produce itaconate and animals that cannot produce itaconate, and we infected with Pseudomonas aeruginosa with the clinical isolate. And we evaluated the airway ketogenesis and hepatic ketogenesis. And as expected, the enrichment of beta-hydroxybutyrate in the airway during the infection with this Pseudomonas isolate was dependent on the ability of the host to produce this itaconate. And also, as expected, the hepatic response was not associated with the production of itaconate. So itaconate is really promoting the enrichment of ketone bodies locally in the airway, but not in the liver. And it looks like these pathogens are exploiting that pathway. We did the same experiment with PO1, and we didn't get the same results. So this is something that the clinical isolate of Pseudomonas have learned how to do during the evolution in the airway of people with cystic fibrosis. So now, coming to the first slide, maybe you're wondering, so do we have itaconate in the airway of people with cystic fibrosis in addition to the ketone bodies? So we quantify itaconate, and we found accumulation of itaconate in this sputum sample. So it looks like this mechanism is already happening there in the airway of these patients. And Pseudomonas are exploiting this mechanism. So just to finish this presentation, now the question is, so what is the local source? Where is this itaconate acting? On which cell type this itaconate is acting to promote this production, these ketone bodies in the airway? What is our local liver, if we can put it like that? So to answer that question, we did a massive metabolotranscriptomic analysis, and we analyzed all the cell types in the airway of these animals infected. Animals are wild type or animals that cannot produce itaconate. And what we found is that if we quantify a ketogenic score, including all the genes that participate in ketogenesis in the mitochondria of cells, we found that only fibroblasts were the ones that activated this transcriptomic program in the presence of itaconate. And so for the people in the audience that really like single cell analysis, here you can see the different genes that are here from the mitochondria that are promoted by itaconids. You can see that, for example, the rate-limiting step gene, that is MHCCS2, it's promoted by itaconid in the presence of Pseudomonas aeruginosa, and also HMGCL, which participate in the production of acetoacetate, and also BDH1 that is here, that participate in the production of beta-hydroxybutyrate. So it looks like this itaconid is promoting a ketogenic response in this local fibroblasts in the airway, and maybe Pseudomonas is exploiting this cell to promote this ketogenic environment and to help the host to activate disease tolerance. And I have much more data that I will not share here because it's too much, but I can show you that, for example, the PO1, this is strain that everyone uses in the laboratory, completely trashes this fibroblasts, and the ketogenic program in this cell, it's gone, it's not there. And that explains why the host response, instead of with the cell, is response with the liver, because that is the classical way how the host will respond to this inflammatory response through ketogenesis. So said that, the conclusion for this presentation, I hope that this was interesting for you, that normally Pseudomonas aeruginosa, when it infects the airway, promotes inflammation and tissue damage. And what we found is that the host activates an hepatic ketogenic response to the infection and mobilizes ketone bodies to the airway to try to suppress this inflammation and the tissue damage. And also this can be modulated by the diet, by the nutrition of the host. So this is something very important to take into consideration. But then in the airway of people with cystic fibrosis, it looks like Pseudomonas learn how to exploit this environment to be able to coexist with the host, and through the induction of itaconid, can promote the same ketogenic program, but locally through the airway fibroblasts. And that will help the bacteria now to be better tolerated and to exploit that lung as a niche to coexist forever with the host. So, and this leads to disease tolerance and the host pathogen coexistence. So said that, I would like to thank my laboratory. And also I would like to thank the laboratory of Dr. Alice Brint, with who we are collaborating constantly, our funding source, the NIH and the Cystic Fibrosis Foundation, and our collaborators. And I will be happy taking any questions that you may have. Thanks very much.
Hey, that was excellent. Joshua Chandler, Emory. First off, I wanted to ask about if I may ask two questions. I want to ask about the sputum specimens first from the donors. How do you, to what links do you go to make sure that the healthy controls are appropriately producing something comparable from lower airways to compare to your CF sputum? I'm sure you evaluate for like salivary contamination, but do you have to go to other lengths to make sure that what you're looking at is really apples to apples? Yeah,
it's complicated to compare, because in the healthy controls, it's in the induced sputum and we compare with the other one. So it's hard to control that. We have, not everything is increasing the CF sputum. So we can like stabilize our values with other metabolites are there. Also we did some analysis of cell counts, and also you can like measure some stuff. And it's a very important question, and it's complicated. That's why then we move into the animal model, just to see if these ketone bodies are there, is really they are being induced, or is it because there is like a bias in the sample. And it looks like it's an intrinsic metabolic response of the host infection.
Thank you. And the follow-up question, or the second question actually, is I was excited to hear about the fibroblasts as like itaconate producers. A lot of times itaconate gets associated with macrophages and monocytes. And I'm curious if you've been sort of looking at relative contributions in the CF airways and maybe other diseases.
So I will clarify that point in terms that we are not claiming that that fibroblasts produce itaconid. It looks like the fibroblasts, they are responding to the itaconid in the environment. Yeah, sorry about that. Maybe I'm excuse myself if I.
No, it was probably
like that.
That's a me problem.
So yeah, we are not saying, but. And I think that we measure RG1 production, which is the gene that produces itaconate, and in fibroblasts, I think it's very low in comparison. It's still there, but it's very low in comparison with the myeloid cells, which are the major source of this itaconate.
Thank you.
Wonderful talk, Sebastian. Katie Heiser, National Jewish Health. So when you got to the end and you talked about how we can modulate our diets and modulate if we're doing ketogenesis, we do have modulators now, but absorption in the CF gut is not normal. And for years, particularly, even with enzymes, fat absorption in particular has been off. And so can you think about your model in the setting of people who can't absorb the right diet necessarily, and how that might be affecting, and maybe it's totally different now than in people, if you were to look at what the lungs look like. And so to that question, was the CF sputum pre-modulator or post-modulator?
It's pre. It's pre mostly. And in terms of the fatty acid absorption, that is a very good question because I thought about it. And in fact, if you don't absorb fatty acids from the diet, the question is how your cells produce energy. So the cells are very smart after evolution and they compensate with other pathways. So if you are not using your mitochondria to produce energy, you will end up switching to glycolysis, which is highly pro-inflammatory. So that can contribute to the hyperinflammatory state of people with cystic fibrosis. So if the modulators, we are not correcting the absorption of fats, our mitochondria still won't be happy and we will still be using glycolysis to that. So I think that we should focus also in helping the host in promoting more, the patient more fatty acid absorption to help the mitochondria to be better in terms of bioenergetics.
It's long been recognized that children who were fed a high-fat diet decades ago had survival advantages in CF. How much variability do you see in the airway metabolites of this depending on the diet that they're fed?
Well, we see difference because with the data that we have with the patient, we don't know the type of diet that we're on. But in terms of the animal model, I can say that we see difference in the ketogenic response, but also we see difference in other metabolites. Okay, so it looks like the diet itself, the ingestion of fats, not only affects the ketogenic response, but maybe also other metabolites as well that are important in immune regulation. And we have some bile acids, we have other metabolites, some amino acids that are they are also accumulating in the airway of ketogenic animals. And we are very curious in knowing how those metabolites also are participating in the immune regulation.
Yeah, it'd be interesting to see if you see those same things in humans as well.
Yeah, of course. It's a good experiment to do, but a little bit.
Hi, great talk, thank you. And I have a curiosity related to the itaconate. In particular, do you think that the mechanism should be local in the lung? I mean, do you think that fibroblasts that produce itaconate can provide an environment only for Pseudomonas? So in other words, do you think that Pseudomonas can be localized close to the cell that produces itaconate in the lung?
Yeah, definitely, yeah. I think that is one of the hypotheses that we have. Back in the past, we published a paper in where we showed that Pseudomonas exploits itaconate to produce biofilms. So I wouldn't be surprised that Pseudomonas thrives better around cells that produce itaconate, such as myeloid cells, and also other type of cells that we'll still know. So I will repeat again, we are not claiming that fibroblasts produce itaconate. It's myeloid cells that produce.
Right. Yeah. And the other curiosity was, do you think that this disease tolerance mechanism can be conserved with other pathogen because the adaptation it has been described for other pathogens?
Definitely. In fact, there are many questions there with other pathogens. Disease tolerance is something that it's known, but we don't understand how it happens. And in immunology, we always think about like inflammation, that we need to survive the infection by clearing the pathogen. But that is not always the case because if we just think about what happens with COVID. We were fighting the infection so much that we end up dying because of the immunopathology. So there are other mechanisms that are important that the host says, okay, I will stop fighting the infection. I will try to survive through another mechanism. And this is tolerant, I think, is important, not only for Pseudomonas, but also for other pathogens.
And have you looked to other CF pathogen for curiosity, like staph or?
Yeah, I think staph, we have data showing that staph also adapts in the airway as Pseudomonas producing biofilms and also suppressing the expression of virulence factors that helps to limit the inflammatory response, which is associated with the tolerance response.
Thank you.
Hi, I'm Marianne Mullerbach. Thank you for this interesting talk. I'm thinking about the human on not modulating their diet, but what contributes to the ketogenesis. So do you think on the hypoxic environment that's right at the airway surface and the neutrophils who eat up oxygen and how that would contribute to things?
That could contribute because hypoxia will be directly affecting the mitochondria and how the mitochondria produce energy through OXPHOS. And normally when you produce ketone bodies, you don't need OXPHOS. You can, so the same hypoxia environment can maybe promote more ketogenesis in their way because the cell, they get deregulated and they try to produce energy through different mechanisms. And by oxidizing fatty acid to produce acetyl-CoA, that does not require oxygen. So maybe it's a mechanism that can get active.
So you think that could be one of the initial drivers for the Pseudomonas to adapt?
Yeah, we don't have data to support that, but as an hypothesis, it's a very interesting hypothesis that maybe we can test. Yeah.
Thank you for the talk. Can I ask you a question with regards to a relationship between systemic ketogenesis and intrinsic ketogenesis that's happening in the airway? Because to my knowledge, you can eat a lot of fat. This is not going to change your ketogenesis, myeloid ketogenesis once the cells are in the airways. And in fact, ketogenesis in myeloid cells improves autophagy and killing of intracellular bacteria. But these bacteria are not going to be easy to take up because they develop a biofilm, right?
Yeah.
So can you comment a little bit on that? Because I think there may be a little bit of a confusion in the field as to whether ketogenesis would change anything in regards to mucosal immunity, but they are, in my view, they are completely separate.
Yeah, it's complicated. I mean, the role of the literature in ketogenesis is pretty broad. It's the effect of ketogenesis can be directly through the fatty acid assimilation, or also can be associated to the microbiome in the gut. And there are many studies showing that if you feed, for example, animals with ketogenic diets, you will change the presence of beta-hydroxybutyrate in the airway. That will change the metabolism of the bacteria in the microbiome, and the bacteria will produce certain short fatty acids like butyrate, and then will change at the same time the production of IL-17 by different immune cells. And those immune cells, they not only locate there, but also they can migrate to other tissues to regulate inflammation and the tissue integrity. So that could be another mechanism, and we didn't take into consideration that mechanism here. But I'm pretty sure that there are many other factors that are also contributing to that, and you are totally right about it.
So I think I'll wrap this up by relating my question way back to the beginning of this workshop, where Juliana had presented data to explain how viruses might promote a secondary bacterial infection in the CF lung. So thinking about your findings or the idea, the concept that you are proposing here, that local production of ketone bodies by fibroblasts might create an anti-inflammatory environment in which Pseudomonas can establish a chronic infection. Are there factors like viral infections that can lead to the production of ketone bodies by fibroblasts? Like, is there a potential connection by which, a stimulus like a virus can then lead to ketone body production in the lung that promotes secondary Pseudomonas?
That is a very interesting question, because I don't know if in fibroblasts themselves, but there are some articles that have shown that ketone bodies are very important during influenza infection, for example. And these ketone bodies, they change the way how gamma delta T cells, they confront the infection. And so I think there is a connection as well with the viral infection. And also there are some data showing that infection with respiratory syncytial virus in epithelial cells induce itaconic production. And also the virus exploit that environment to persist a little bit more, because the bacteria, as itaconic is anti-inflammatory, the cells, they stop fighting the infection, and it looks like the virus takes advantage of that. But that is in epithelial cells. I don't think that there is data with fibroblasts yet.
Much more to learn. So thanks, Sebastian. That wraps up our workshop.